In this video, I'll be showing you how to answer question 2 of the second summer assignment, and it's right from your textbook, Concept Check 14.1. And with that question, you're going to need Table 14.1, which is given to you in my video right here, along with your textbook. So what I'm trying to do here in this video is give you some hints and show you how I would complete this question since I'm not in class to show you live. By me modeling this question, it is my hope that you'll be able to pick up the necessary skills to answer the question 2A, which will not have any video associated with it except this one. So by watching me complete this question, I'm modeling how you should complete the second question. It'll be similar but not the same. Notice that if you don't like me reviewing this question, okay, that you're doing, you could also go to the Appendix A and see their explanation. Although I think maybe me walking you through it step by step might be a better plan for you. Okay, so let's get started. So number one, pea plants, heterozygous for flower position and stem length. And there they're giving you the genotype of the alleles that we now know are located on chromosomes, are allowed to self-pollinate. Okay, so keywords here, heterozygous, and they're giving me the genotype, okay, which will give you the phenotype. Now, phenotype is the actual traits that we see, all right, are allowed to self-pollinate. That's an important word because now we've got this set of alleles from chromosomes going to basically be crossed with another set of the same type. All right. Now, how did that occur? Well, probably it was occurred from having a P1 generation, the parents, and the parents were probably true breeding. Just to go over that because you may see this in the other question. So I'm setting this up for you. So probably what happened is they took something that was homozygous for these traits, true breeding, okay, AA and TT. Now, what does AA and TT mean? Well, flower position, okay, you see the big A. So we go to our 14.1 table. It's very important we know how to use information given to us in this course. So we go to flower position. Notice it says axial. What does that mean? That means the flower, okay, has flower position is in the axial plane down here, okay? Now, notice in the chart, the dominant trait is listed above. So they're telling you axial is dominant. So this big A, okay, you should realize means that we're dealing with the axial position of the flowers in the pea plant, and it's a dominant trait. Notice the terminal, okay? Terminal means the flowers are at the end of the flower on top here. That's their designation. Now, it's a recessive trait, so that would be little a, okay? So little a is the recessive trait there, okay? Notice if I'm starting with the parental true breeding, all right, I wouldn't have the recessive trait at all in this organism that carries these alleles, which again are on chromosomes. Okay, now I'm going to cross this, okay, with another true breeding. Oh, I'm sorry, I didn't finish. I had AA, which means I had two dominant homozygous alleles for the axial position of the flowers. And TT, well, they told me that I have stem length also involved here. So stem length, okay, I have to scroll down. Oops, scrolling down, I see stem length, okay, is tall, and that's going to be in the dominant position, and dwarf is in the recessive position. So what they used, T, for the tall stem length, okay? So in order to set this problem up, really, how do they come up with this AT? I'm just showing you how they would do this normally, and that's what the second part 2A question kind of starts you with, so you understand. So in order for me to get to this heterozygous for both traits, what they would do is they would take the parental generation, they would take a true breeding, AATT, homozygous for both dominant alleles, and they would cross it with heterozygous for both alleles. Okay, sometimes they'd call this double mutant, but this is double recessive. It doesn't matter. The little a, okay, as we learned, is the recessive gene for the axial position. That's the terminal. And the little t, okay, is the recessive gene for the stem length, which is dwarf. Now, we should all recognize that even though we're doing Mendelian genetics, which is very simplified because he dealt with a pea plant with 14 chromosomes, and thankfully in his work he picked something where these genes were on separate chromosomes, okay, so they weren't linked in any way, but he was lucky in the fact that he picked a plant that was simple enough to come out with the crosses and the probabilities that he expected using his two basic philosophies developed: the law of segregation, the law of independent assortment, which I'll go over here. But any case, when you cross these together, okay, if you do a simple Punnett square, you will see that we're going to come up with the F1 generation, okay. I think F is phyla for son, so basically these are the offspring. What you can clearly see is we're going to come up with A, little a, t, and little t. And you should be able to see that. If I was mixing these chromosomes, that's what you would have, okay? You remember, you would basically break these down and can see that mixing these A's with these AA's. And if you need to see this, make yourself a small little Punnett square. What I mean by that is, assuming that these A's break apart and segregate into gametes, into different eggs or sperm, these would break apart and these would break apart. That's called a law of segregation. So if you don't see how I came up with this as the only genotype from the cross between these true breeding, now I say true breeding again because they have complete, okay, homozygous for their traits or their alleles. Okay, they're not carriers, not heterozygous. So if I was to make a simple Punnett square, I would have A, little a, big A, little a, big A, little a, big A, little a. So just putting together these two alleles, okay, and crossing them without considering the T's, you can see that all of my genotype would be the same, which is A, little a. And if I was to do the same and break apart the T's, they would segregate, law of segregation, into different gametes, okay? You would have what? You would have little t and little t from these guys separating, you know, and we could make this the maternal and this is the paternal. So this would be the egg. This would be the sperm, let's say. Doesn't matter. What are you going to get? You can see that you're going to get the same heterozygous relationship. And that's when you read the book, guys. You really got to do more than just to read. You've got to understand. You've got to go through some of these problems as well. And that's what I'm forcing you to do here. So clearly you can see by just segregating these, breaking apart these alleles, jumping into their gametes, which we call the law of segregation, we can see that we would get one offspring, okay, at least an offspring of the same genotype and phenotype in this matter. Notice they'd be, in this case, be carriers of the recessive allele, but because they're dominant genes and recessive genes, not co-dominant here, okay, you would have the A, which is the axial phenotype being shown, and you would have the terminal, the tall, okay, stem plants phenotype being shown here because of the dominant allele, okay? Now, a little reminder, recessive alleles, okay, in this more simplistic way, and it is true in some of the genetic disorders, recessive genes sometimes are broken genes or genes that do not code for any kind of protein that works. So therefore, if it's heterozygous, and like in this case, the dominant gene is the gene that codes for a specific protein that makes the stem be a certain height or give the flowers in a certain area, whereas the other gene's just not working because of, let's say, a mutation. Okay, and that's something you should truly understand. Before I continue with the problem, though, let's talk about how we can deal and incorporate this chromosomal type of inheritance. Remember, Mendel did not consider chromosomes, didn't even know what genes were. He was just looking at these patterns of inheritable traits and trying to figure out through his Punnett squares, but he definitely came up with two laws that are still true today. So we can see at this moment, all right, that we have made, again, completely big A, little a, big T, little t offspring, and I got all of them. Now, that's where the problem really starts, and that's where I wanted to make sure you understood. In order to get to this point, we had to take, okay, a parental true breeding for both, and you'll see this in many studies, and they'll make, okay, this heterozygote offspring, where again, we are dominant for the axial and dominant for the tall, but we're carrying the recessive trait. Sometimes we call it carriers. And we should recognize the fact that the recessive allele at this moment is normally broken alleles. These are alleles that code for, or these are genes on chromosomes. Remember, it's connected to a chromosome where we are coding for a gene that's not working. So if you are heterozygous, you have a chromosome that's coding for some kind of protein or enzyme important in the synthesis or construction of the plant at some point that makes it have a flower at a certain position or have a certain length of stem. Okay, the recessive allele usually means that there's a mutation in that base pair of the DNA that's on the chromosome. Okay, so Mendelian genetics opened the door for us to understand. Number one, we're lucky because he picked something that only had distinct traits that we can see on different chromosomes. It's important you get that. So he was lucky in that sense, but his work opened the door to be able to use genetics at a more advanced level. But in any case, these traits, luckily, okay, are on different chromosomes. And because of that, he was able to come up with good mathematics to help us understand the probabilities of attaining these outcomes. Or at least he saw patterns that made him understand and come up with two very important laws. Okay, but what the problem starts with, and I'll pick that up in a second, is he said in this problem, or in the problem they wrote, the authors did, Mendel didn't, they said if you start with this, now of course I overtaught this and showed you how they got to this position by doing true breeding crosses, but now that you have the heterozygote for the two dominants, they're self-pollinating. Now they want to know how many will have terminal flowers and be dwarf. So they're looking for a certain phenotype that we're going to get from the genotype in this cross, but we're going to cross heterozygotes. So we're basically going to do is we're going to cross the offspring. If they're going to self-pollinate, what we have is we have the homozygote doing what? Okay, crossing with itself. Okay, and we call this something. This is called a dihybrid cross. And you saw this. Why is it a hybrid? Because we have added the recessive character to both. It's di because you're using two different alleles now. All right, even though I did individuals so you can see that. So now what this is about is doing a dihybrid cross. Now before we do that, we have to understand what are the gametes here. Remember, Mendel used these very simple traits and saw patterns in his work that led him to two basic laws. The law of segregation. Now, segregation means, as we've talked about, that these alleles that are now we found on different chromosomes separate and go to different gametes. Okay, so what are the possibilities here? Let's think about this. Okay, well, the possibilities are that, well, let's let's write them on a chromosome so we can see it. We have basically 4 chromosomes here. So I'm going to write them. Now, pea plants, I believe, have 14 chromosome pairs. So I'm just going to write this in terms of a haploid cell, diploid cell, with 2n equals 4 chromosomes. Okay, so what am I going to do here? Well, here's A. A is going to be my black. Here's an A. And here's little a. And we'll do red for the T. All right, so AA is going to react with TT. Well, it's not reacting, this is one cell, so I should make that very clear. So this is T and then little t. So you can see. And what I'm showing you, something called loci. This means that I'm showing a particular location on the chromosome. So when we write AT, we're really saying that there is 2 positions on a chromosome where this part of the DNA in the chromosome codes for an enzyme that makes this phenotype, which in this case, the axial place where flowers are, or the tall stems. All right, that makes the phenotype. So it's the genotype that makes the phenotype. From the phenotype, which is what we see, we can, guess what the genes are, dominant or codominant, on the chromosomes. And you'll hear me say the word alleles. And you can say that the expressed gene on a chromosome is an allele. So these are 2 alleles, okay? Any case, how does this work? Well, through meiosis, because of the law of segregation, what's going to happen is we're going to pull apart these chromosomes. So we're going to pull apart A, and it's going to go to one gamete, okay, and we're going to pull apart another one, and it'll go to another gamete. So these 2 can come together. And we call that independent assortment. Now there's 2 parts. The law of segregation means that these 2, I should say, these 2 are going to split, okay, and go into different gametes. And then if you have a second trait that we're looking at, that these traits, okay, will independently separate from the others. So basically, the law of segregation and independent assortment, and I'll write it, are about the idea that these traits somehow split and make half of the information from a diploid cell. They do so totally randomly and because they do so, we can use probability rules. So when I put this together, okay, and let's make a new possibility. So if these 2 were to come together, that would give me a chromosome pair with an A and a T, and a T is, I guess I'm using red, a little t in this case. This is one possible arrangement. Notice now I'm dealing with not a 2n, but an n. So this is going to be n equals 2. Each gamete cell will have the number. Remember, when you made a cell, this is an entire cell, 1 came from the mother, 1 came from the father, 1 came from the mother, 1 came from the father. Now we don't know which one is which. We certainly could trace that in something called a pedigree chart, but we're not doing that here. So all we're doing is saying these guys are separating and segregating, and they're independently assorting into each gamete. So this is one possibility. Well, what's another possibility? Okay, we did the first and third. What's another equally possibility here? How about the inner 2? How about these 2 would separate, segregate in anaphase of meiosis, and of course come and make a gamete. So we would have, let's do this one. This pair would be, there's one chromosome, here's another chromosome, and we'd have our, what, little a and we would have our, go red here, big T. So we did in and out, kind of like we're foiling here. What's another possibility? How about outside, inside, or the first and third? That would give me this pair of gametes. Remember, either one of these possibilities is a complete chance, equal chance here. Okay, so if I go the first, they give me big A on this chromosome, and then it'd be big T on this chromosome. And then I'll draw the next pair, since I'm in blue, or gonna get blue. All right, so if you don't like that drawing, too bad. So we did first and third. I think second and fourth is my last combination. And what do I have? I have little a. Going to the red. Got the little t. And there are no more possibilities. Okay, so these are the 4 different possibilities in making gametes. Remember, this would be we're going from 4 chromosomes down to 2 in the meiotic halving here. Okay, in meiosis 2, actually, that's where it occurs. But the point being is that these are all the possible or possibilities that I would get from doing a dihybrid cross of 2 traits. Okay, we're also making the assumption that the traits are not linked. They're on different chromosomes. It's the only way that Mendel was able to come up with this independent assortment law. So segregation, gametes split from each other from one trait into different gametes. Independent assort means this happens if you have multiple traits. All these traits separate from each other independently. And what we get is we get 4 possible outcomes. And because of those 4 possible outcomes, this could happen 1/4 of the time. This can happen 1/4 of the time. This could happen 1/4 of the time. These are 4 mutually exclusive possibilities. Now you'll learn something about mathematics, and we have to bring mathematics into this to understand patterns and understand what's happening. And that's what Mendel did. He understood that mathematics was an important application of his studies. Now, using probability and using these rules based upon these individual chromosomes or alleles on information on the chromosomes would separate based on total probability, okay, then you realize that I can take 1/4 and I can add it to 1/4 and I can add it to 1/4 and add it to 1/4. What I'm doing here is I'm adding all of these individual different outcomes that are mutually exclusive, okay? And you can see that, of course, I would get 1. So this is all the possible outcomes that I can get if I'm dealing with a cross between a mother and a father that have these alleles for those genes, or those genes, right? So we call mutually exclusive rule the addition rule. Now you say, well, why am I excluding? Well, there's only these 4 outcomes. There's nothing else. We exclude everything else because these are the only 4 possibilities. When you say mutually exclusive, when you're doing the reading, hope you thought about that. That means you either have, in case of a flipping of a coin, heads or tails. There's only 2 possible outcomes. That's why if you were going to do 1 toss, it would take what? You'd have a 50% chance of getting half. And if you tossed it again, you'd have a, well, let's stay with that. If in 1 toss, 1/2 of your outcomes is heads, the other half is tails. And you add them together, that will equal a complete 1, all of the outcomes. That's what I'm doing here. So the reason why we can use 1/4 is because of the idea that we're either heads or tails. We're either going to be this combination or this combination. It's either going to be these 2 coming together or these 2 or the other 4 possibilities. Okay, there's no other possibilities and they all equal to 1. That's important going forward. Okay, so let's go on to our Punnett square, which is basically what this is about, but you needed to understand that. Okay, so let's just draw our Punnett square. Let's put out all the possibilities again from the last page. We had 1 gamete that could be AT. We had 1 gamete that could be capital A. and little t. And then we had another gamete that could be A and T. Notice we started with 4 chromosomes. Now we're down to 2 chromosomes, or 2 alleles. Okay, and then we have A little t. And we could say this is from the mother. This could be that this is from the eggs, let's say. Okay, and you'll see this many ways. And this could be from the sperm or the dad. It doesn't matter because this is a dihybrid cross. In a dihybrid cross, you have the same genotype. So we're going to have, if you were to make, let's go back. We're taking this, remember, this is one complete cell. This, let's say, is the mother. The mother would break apart into 4 possibilities. Since the father has the same genotype, it would make the 4 of the same gametes, okay, in meiosis. So whether it's the mother or the father, you have the same combinations. So in a dihybrid cross, that's why we do this, make life a little easier. We're going to have the 4 same. So these are the 4 different combinations that we just got from above here. Okay, that's what this is. And if we did this for the, let's say, the mother, the eggs, then the father with the same genotype would also produce sperm or gametes, okay, with the same combinations or the same 4 choices, okay, or possibilities based on law of segregation, the law of independent assortment. Okay, so these are our gametes. Okay, and now I'm just going to make a line, and let's see if I can do that. Not bad. Okay, that one just got crazy. Now let's put these guys together. Okay. And let me just move this line back because I don't want to be too tall in this process. Boy, that stinks. Here, and then I come down. Okay. All right, so let's do our cross here. And what am I going to get? I'm going to get 2 A's and 2 T's here. 2 big A's, 2 T's. That's going to be the, if these 2 gametes from each of these eggs or sperm from same dihybrid cross would make this genotype. Okay, then I'd have big A, big A, big T, little t. Big A, little a. And this becomes almost fun. Now maybe not T, T. Big A, little a. Big T, little t. Let me keep going down. Now this one's going to cross with this one. AT, so big A, homozygous for the axial, big T, little t. And we just keep going. You can see what I'm doing, hopefully. In this case, we have a recessive allele. Okay, the dwarf, we're going to have a dwarf phenotype because of that. Okay, and then we have A, little a, big A here, little a here. And then big T, the dominant always goes first. Okay, A, big A, little a, and then again, ooh, another recessive allele, 2 little t's. Okay, little a, big A. Again, I'll put the dominant first, and then 2 big T's. Big A, little a, heterozygous for flower position. Big T, little t. A, little a here, so now we're recessive, okay, for the flower position. That's going to be terminal flower positions, and then big T, little a, big T, little t. At this point, I hope you can get what I'm doing. A, little a, T, little t, big A, little a, 2 little t's, 2 little a's, recessive for the position of the flowers, big T, little t, and then recessive for both. Okay, and remember the question So these are the offspring. This would be the F2 generation from the dihybrid cross if we started from true breeding. And they said, how many offspring predicted to have terminal flowers and be dwarf? Well, terminal flowers, okay, if you remember, is the recessive gene. That's the little t. So we would have to have little t, and dwarf flowers. Little t, my bad. Terminal flowers, because we're starting with the A for the axial, would be little a and little a. And then dwarf flowers would be little t. So both recessive, okay, alleles need to be there for the recessive phenotype to be shown. And there we go. So they're asking, well, how many? Well, you can clearly see 1, 2, 3, 4, 1, 2, 3, 4. There are 16 possible outcomes. So 4 times 4, if you want to add them up, you can. You're going to see 16 possible outcomes of genotypes, different types of alleles from the different combinations of what? Chromosome pairs with each other. And you can see 1 out of the 16 will have the recessive, okay, gene being expressed, have the phenotype. Only 1 out of 16 will have the phenotype of terminal and dwarf together. And because they said I have 400 seeds are planted, how many would grow? So if these represented, if you had 400 seeds and 1/6 of your offspring is going to have this genotype, all right, then you need 1/16 out of 400. For those that are not new to the math, all you do here is take your 400 and you would times it by 1/16. And that would give you how many? That value, of course, put in your calculator, you get 25. So 25 plants out of 400, okay, will have the recessive gene being expressed, okay? Now, what I need you to understand is I could look at this a little differently. I could actually solve for this mathematically, and this is what the other problem, number 3a, is going to be like. Think with me for a second. We did the addition rule. You know, if we're looking at gametes, you're either going to have, these were all what? Each one of these gametes were a 1/4 possibility. There are 1 of 4 outcomes that could have been made when I split up the each parent's alleles or chromosomes up. So if you think about this, 1/4 for each. Now, if again you don't like that, you can do Punnett squares individual to see that, but there's 1/4. And again, if you don't understand what I'm saying, the 1/4 is coming from the 4 possibilities of each of the mother's and the father's gametes. So my friends, I could have solved this without doing much of a Punnett square. I could have done it this way. I can say, well, take 1/4 of one gamete, and I could times it by 1/4 of another gamete from, this is the mother, this is the father, and we get 1/16. So I could have came up with this without doing the Punnett square, and that's what most people do. Now, why am I multiplying and not adding? When you add, like a heads or tail on a coin, this is the president's face, okay? That'd be a Lincoln, I guess. You get heads or tails, and all the outcomes you add up to equal 0. 1/2 of this, you have 2 sides of the coin, hopefully, and when you add it to another half of the side, you get a total of 1. Well, you had what? 1/4 possibilities for all the gametes to be made by pulling the chromosomes together, and you get 1. When you have events that are totally independent, that means a sperm from one father hits a mother. These did not come from the same chromosome. Okay, we multiply. If I'm going to flip a coin twice, what would I do? And what would the chances of getting 2 heads? Well, think with me for a second. If I'm going to flip a coin twice, The first possibility to get a heads is a half. The second is a half. We multiply, you would get 1/4. So 25% of the time, remember 1/4 is 25%, is how you get 2 heads. Mr. Grotsky, how does that work? Well, when you multiply, you're dealing with 2 independent events. Pulling apart gametes from one cell is not independent events. They're tied to each other. Flipping a coin twice is not. A sperm hitting an egg is not. Okay? These are independent actions. When you multiply, you get 1/4. So when you take 1/4 times 1/4, you get 1/16, and then we would times it by the 400. Now, if you still don't quite know why multiplying would be used, think of the outcomes. When I flip a coin, I can get heads. I can get what? Tails. And then I can get heads or tails. How many outcomes are there? 4. So 1/2 gives you the 4th. Now, why is there only 1 on top? Well, I can get heads, heads, tail, heads, heads, tail, or tail, tails. Isn't there only 1 outcome out of 4? So multiplying is used when we do these Punnett squares where we're taking gametes of one, hitting gametes of another. There you multiply because they're not tied to one single cell. They're tied to different independent actions. And that's what number 3A is going to be about. But the answer here is 400. Yes, I overtaught this, but I want you to understand the underlying connection between the chromosomes, Mendelian genetics, and his laws that are still applicable today. Hope that helped.